to present to you our research results about the effect of aleurone supplementation to horses in training. I'm both affiliated to Ghent University Research Group of Comparative Physiology and the Equine Hospital in Wolvega. First, I will give you a small recap of what aleurone actually is. So aleurone resides in the outer bran layer of the wheat kernel. And as you can see in the upper right picture, it surrounds the starchy endosperm and the germ. During milling, it stays with the hyaline layer and the miller considers it part of the bran fraction. In the budding wheat kernel, it supplies proteins and signals to the growing wheat kernel. Aleurone is thought to be responsible for the positive effects of whole grain products on gut and metabolic health, and several scientific studies have been executed in pigs, rats, and recently also in human. The aleurone layer is particularly rich in fiber, minerals, nutrients, and bioactive phytochemicals, the osmolyte betaine and the antioxidant ferulic acid. And the problem was always to be able to free all these substances from the fiber layers, called the arabinoxylan layer. But new milling techniques have increased the biological availability of the bioactive substances from aleurone, and that has stimulated a lot of recent research, including our own. So in 2017, we presented results from a previously executed resting trial at the ECIM Congress. And in this study, we examined which dose of aleurone would be beneficial for oral supplementation in horses. So studies on the effects of aleurone on health have been focusing on antioxidant function, the responsible metabolite for that being ferulic acid, and changes in glucose and insulin dynamics caused by aleurone have been found in other species before as well. In this study, pigs had a lower insulin secretion when fed an aleurone-enriched diet for 4 days. And it is also known that betaine insufficiency is associated with lipid metabolism disorders such as diabetes and metabolic syndrome. And a recent publication about obese mice with hepatic liver disease, in which in this publication the mice were fed aleurone, and this decreased the triglyceride content in their livers, changing the energy management of these obese mice. It was also shown that aleurone induced a higher alpha and beta diversity in the gut microbiome of these mice as well. To study the effect of aleurone supplementation on horses in training, we performed a crossover training trial with 16 horses that were trained 2 periods of 8 weeks, of which 1 training period with aleurone supplementation and 1 period being fed a isoenergetic diet without aleurone. Before and after each training period, a frequently sampled intravenous glucose tolerance test and an in-feed oral glucose tolerance test were performed, and fecal microbiome samples were taken. We purchased 16 standardbred mares, all 3 to 4 year olds, that were all housed and trained at the same facility by the same trainer at the same racetrack to maximize standardization for this trial. So we aim to apply a mechanistic physiological approach and analyze several parameters in muscle biopsies, in BALs, and in blood. So there are many more results to come, but for now we will focus on the effect of aleurone on glucose-insulin dynamics and the gut microbiome. So in the pre- and post-training weeks, postprandial blood glucose and insulin curves were constructed after an in-feed OGTT, in which 500 mg per kg glucose was provided. And on a separate day, we performed a frequently sampled intravenous glucose tolerance test in which 300 mg per kg glucose was provided. And for the blood sampling schedule, you can refer to the table on the right of this slide. Standard parameters were included, such as maximum glucose and insulin during in-feed OGTT, and area under the curve for glucose and insulin, and time to peak insulin. For the frequently sampled IGTT, a minimal model analysis was performed using MinMod software, and outcome parameters included acute insulin response to glucose, insulin sensitivity, glucose effectiveness, and disposition index. To study the fecal microbiome, the V3-V4 region was amplified by PCR and sequenced. Microbiome composition was analyzed in KIME2, and to check if the microbiome composition would enrich for certain microbial energy pathways, we ran PyCrust using the MetaPsych data. These are the statistical analysis that we used. And in addition to this, the data in the microbiome analysis was Bonferroni corrected. So adding aleurone did not really change the OGTT parameters, but a trend was shown for time to peak insulin when combined with training. But this was not significant. Training did significantly decrease the maximum insulin levels and the area under the curve insulin and increased time to peak of insulin. No changes in glucose levels were found here. When applying the frequently sampled IGTT, we did find significant changes when feeding aleurone to horses. A decreased acute insulin response to glucose was found, the AIRG, which is a measurement of the fast and first response of the body to IV glucose, how much insulin is secreted in the first period after IV glucose dosing. When looking at more insulin-resistant individuals, they will have higher AIRGs. The second parameter that significantly changed by adding aleurone to the diet was an increase in glucose effectiveness, the SG. That is the insulin-independent glucose disposal. It is glucose-driven uptake of glucose into the tissues, such as skeletal muscle and the liver. So in short, this is the capacity of the body to take up glucose from the bloodstream without insulin or at basal insulin levels. And if we compared the frequently sampled IGTT results from week 8 with aleurone versus week 8 without aleurone supplementation, there was also a significantly lower acute insulin response to glucose in the aleurone-fed group. Aleurone supplementation increased the beta diversity of the fecal microbiome significantly in our study. And aleurone significantly decreased the bacterial genus De Sulfovibrio. Training alone without aleurone increased the genus Pseudomonas and decreased the Streptococcus genus. So to check if the microbiome composition changes would enrich for certain bacterial energy pathways, we ran PyCrust, and the resulting relative pathway abundances were compared between trained with aleurone versus trained without aleurone. And here you can see, for instance, that adding aleurone to the diet increased the aromatic amino acid degradation in the bacterial microbiome. However, this is just a prediction, and we are currently looking into the metabolomics of these fecal samples to see if we can confirm these results. We also looked into correlations between the glucose-insulin parameters and the microbiome changes caused by aleurone. So two of the Firmicutes Clostridialis families, namely the Peptostreptococcaceae and the Lachnospiraceae genus Marvin-Briantia, show a strong and significant positive correlation with the changes in AIRG caused by aleurone supplementation. So the acute insulin response to glucose was lower after aleurone supplementation, and this is correlated to also less peptose and less Marvin-Briantiae. A strong negative correlation was found between the differences in glucose effectiveness, the SG, induced by aleurone supplementation and the relative abundance of the bacterial family of Moraxellaceae. So adding aleurone to the diet increased the capacity of cells to take up glucose at basal insulin levels, which is the SG, and this is strongly correlated to decreased abundancy of Moraxellaceae families. So aleurone increased peripheral glucose uptake in trained horses, which was shown by the changes in glucose effectiveness and AIRG. And possible explanations for this are that aleurone leads to, for instance, an increased number of glucose transporters on the cell surface, which we are actually trying to analyze at the moment using Western blot for GLUT4, 8, and 12. And to further clarify what aleurone and training change in the skeletal muscle, untargeted metabolomics are also being performed, and results of this will follow. De Sulfovibrio decreased significantly when adding aleurone to the diet of trained horses, and in multiple human studies, this bacterial genus is increased in humans with diabetes type 2. We predicted changes in microbial metabolite production caused by aleurone, and we are performing a fecal metabolomic study at the moment to see if these predicted changes are true. Aleurone supplementation was beneficial for glucose-mediated glucose disposal, and this is correlated to microbial flora changes. How can we explain this? What is the connection between adding aleurone to the diet and the healthier glycemic parameters that we found? Hopefully, fecal metabolomic studies will further elucidate this. So to conclude, aleurone had an additional beneficial effect on glucose and insulin dynamics on top of training in healthy horses, and this was attributable to increased tissue glucose uptake capacity. Aleurone increased the beta diversity of the fecal microbiome in trained horses, but there is, as always, more research needed to elucidate the mode of action of aleurone at the level of the skeletal muscle and the gut. Thank you very much for your attention, and I'm happy to answer any questions.